you, everyone.
All right, thank you so much. Our final talk for the afternoon will come from Adam Glassman. Adam is the Executive Director at the JABE Center for Health Research and serves as the JPI for the DRCR Retinal Network. And he'll be talking about clinical trial considerations in endpoint validation.
Excellent, thank you. So right after this talk, we're going to break again into the breakout groups, and we're going to talk about designing clinical trials based on some of the conversations that we had this morning. And so I'm going to talk just a little bit about some of the topics that we might want to consider while designing those trials, clinical studies. No conflict of interest. So, you know, as we saw this morning, and from those quick hitting 5-minute recap sessions, there are a lot of clinical endpoints that we have to consider. And just a little bit of a recap, you know, this morning we talked about prioritization of these clinical endpoints, and specifically, you know, we narrow them down to 2 large bins: clinical endpoints and surrogate endpoints, where clinical endpoints directly measure how a patient feels or functions or survives. You know, we talked about, for example, in visual function group, visual acuity, not necessarily, as we're looking for new clinical endpoints, not things that necessarily correlate with visual acuity, but things that are measuring something different. Surrogate endpoints, on the other hand, are a substitute for directly measuring these clinical endpoints. They're useful when clinical endpoints might take a long time to study, but we do need, as has already been mentioned, we do need extensive evidence from the trial showing that a surrogate can be a predictor of a correlate, and the FDA does have some guidance on how to do that. As we consider different endpoints and their applicability to either medical care, clinical research, regulatory approval, some of the discussions and the decisions on trial design may be different depending on its applicability. And certainly, as we talked about this morning, the discussions vary pretty greatly depending on disease stage. Okay. So what is needed from clinical studies towards this goal of endpoint validation? And really, there's 4 kind of bins that I'm going to briefly touch on today. Characteristics of measurement, association with disease, logistics, and then briefly on statistical considerations. So measurement, characteristics of the measurement. So really, even before the study starts, we do need to understand the properties of the tool that we're ultimately going to be used to measure the outcome. You know, is it measuring what we think it's measuring? How accurate is it? Is it objective? Certainly, the more objective, the better. And then once we're comfortable that the tool is measuring what we think it is, then we need to start collecting more data and understanding the results of how the tool can actually be used. So within a study, several characteristics of the measurement tool that is going to be important for us to understand: reproducibility, test, retest, intersession, intergrader, depending on how subjective or objective the measure is, and certainly we'll do this by getting repeat measures on the same eye, understanding learning effects or fatigue effects, and the measurement range, which is not necessarily just an aspect of the machine itself, but also could certainly be based on the cohort under study. And so here's just a quick example of some limited data from contrast sensitivity. I'll use some contrast sensitivity examples throughout the presentation. And so as you can see here, you know, we have a pretty small range of contrast sensitivity outcomes and a very small number. Only 43 patients went into this reproducibility data. So much more data is needed so we can start to understand what some of the characteristics of contrast sensitivity are, and specifically whether a change is beyond what we would expect from measurement variability. So here is OCT, a study that DRCR did years ago. We took hundreds of replicate measures, spectralis OCT, and so now we have a pretty good sense of what defines a real change on OCT. You can see the dashed lines there right around 25 microns. And so we're pretty confident that anything over a 25 micron change is real. Whether it's clinically meaningful or not, that's a different conversation, but it's likely outside of the range of what we would expect for variability. It's a pretty objective measure, so we also see a pretty good distribution around 0, so there's not much of a learning or fatigue effect we wouldn't expect. It's, like I said, it's a pretty objective measure. But we can learn a lot, but it does take a lot of patients to get a really good handle on those characteristics. Okay. So the next bin I want to talk about is association with disease. And here I'm going to touch on 3 topics: correlation with disease, sensitivity to change, and meaningfulness. So first, probably the easiest, if you will, of those is just correlation with disease. And so this very simply is just the correlation of the clinical endpoint with the disease stage or the duration. It's usually measured cross-sectionally at a single time point, and we compare the endpoint with the disease duration or whatever the other outcome of disease severity is. And as an example, again, going back to contrast sensitivity, we can see just looking cross-sectionally, again, relatively small n's, but when we look at a variety of disease states, all the way from no diabetes through several stages of diabetic retinopathy, we can see that the sensitivities do worsen throughout the course of the disease process. So that's just cross-sectionally, not that complex. When we get a little bit more complicated, we look at change over time. And so this is more challenging because we obviously need a much longer study. We're looking at how the disease changes compared with how the endpoint changes. And so this is usually measured longitudinally. How long is going to really depend on what we would expect some of the natural history of the disease to experience. And so that will be part of the discussions that we have later on. And not unexpectedly, we want to compare the changes with the endpoint with the changes in disease severity. And this does factor back with understanding how to detect a real change. And again, sort of correlated with understanding the measurement itself and what really does constitute a real change. And so there's limited data, going back to contrast sensitivity example, limited data from contrast sensitivity. This is actually from RISE and RIDE in a population of DME patients. And we look at, on the x-axis, change in DRSS with mean change in contrast sensitivity on the y-axis. So there's not a lot of data there. Where this has been used outside of diabetic retinopathy, but within diabetes, is actually a relatively new outcome that has gained a lot of popularity, percent time in range of glucose measures. And so here you can see on the x-axis the percent that an individual is in range with their glucose measurements, and on the y-axis, bad outcomes, either frequency of retinopathy, frequency of kidney disease. And as you can see, as percent time in range is lower and lower, which is obviously bad, the increase in the prevalence of each of these different diseases. All right, so the next final aspect of this group is clinically meaningfulness, and this is really, really challenging. We've already talked about it. Directly measures how a person feels, functions, or survives, looking for clinically important differences in the individual and in the cohort, and really trying to understand if it's a change, not just is it beyond reproducibility, but does it actually impact how the person is experiencing life. Going to logistics, currently we're doing a study with visual fields, Humphrey visual fields, and this is sort of a perfect example of all the challenges that can come up with logistics. Many of the clinical sites, the retina practices that we have, do not even have this machine, so availability and cost certainly are an issue. It takes quite a long time to do. There are challenges depending on body type and age just to obtain Humphrey visual fields. And even before we started, despite some of the, using a lot of the glaucoma experiences to standardize methods with written procedures, find skilled people, particularly within retina practices that can do these sort of things is a major challenge. As we look to statistical considerations, defining the outcome, whether it was continuous or time to event, and thinking about all the pros and cons to binary versus continuous outcomes will be critical. And so clinical trials are going to be needed for endpoint validation. Obviously, the cross-sectional studies will provide useful information, but we really do need those longitudinal studies. Endpoints have to be measurable and reproducible and associated with disease. And the sooner we can start prioritizing these endpoints into studies, the better off we'll be. And thank you.